Hi, so I'm Dr. Amanda Hagee. I am in the Department of Biomedical Sciences at the University of North Dakota, and today I'm going to show you how to passage your cells in a biosafety level 2 hood here. So with these kind of cell culture hoods, what you want to do before you use them is make sure that you turn them on ahead of time. It gives them 5 minutes to kind of blow and become a sterile environment for you. Your hood should also be fairly empty, particularly between the front and the back, to give yourself a consistently sterile field. The first thing we're going to do after our hood has been on for 5 minutes, and to turn it on, usually there are various light switchers. You may have a blower fan to turn on and off. And then you raise the sash of the hood to the line indicated by the sticker on the side. Before we get started, we want to spray down the hood, and so we're going to be spraying down everything that goes into the hood with 70% ethanol to maintain the sterility in there, including your hands. So anytime in question, whether you've touched your glasses or your hair or anything like that, you want to make sure that you respray your hands with ethanol to maintain the sterility. And then I'm in, obviously, lab coat, gloves, and normal lab attire, long pants, and closed-toed shoes. And then usually you just use a chem wipe to wipe down the ethanol. For passaging cells, we're going to have to spray in multiple items that we're going to use. So one of them is going to be your either automatic pipette, pipetteman, there's several different names people use for them, but a pipette that then uses long serological pipettes, somewhere between 1 mil to 25 mil pipettes that look like these. So again, everything that goes in the hood gets sprayed down. I'm also going to be spraying in, these are glass Pasture pipettes. They're what we use with our aspirating system. So there may be many different variations of this in the lab, but essentially there's going to be some form of pipette tip to go with your vacuum pump, which we'll use to aspirate off media, which you'll see throughout the process. Then we're also going to be spraying in the various medias and solutions that we're going to be using to passage the cells. Today that's going to consist of, we have 1X PBS, we have our cell culture media. It's generally always going to be pink. Before you use any of your cell culture media, you want to make sure and check that there are nothing floating in the media and that it is a consistent pink color versus being yellow or having several things floating in it or any sort of opaqueness to it. It should be translucent. And all of these solutions were in a 37-degree water bath before use, so that they're the same temperature of what our cells are in the incubator. That's also going to vary a little bit on cell type, on how necessary that is. Some cells are more sensitive to temperature changes, while some aren't. We're also spraying in our bottle of trypsin. The concentration, again, is going to be dependent on your application. It's going to be what we use to lift the cells off of the plastic dishes that they are on. And then I'm also today spraying in a bottle of 1% gelatin for this. So this is going to be something that we're going to treat the dishes with. This is something that's used primarily for primary cells. So things that were directly derived, say, from a mouse or possibly a tumor human samples, those kind of things. It's just really dependent on your cell type, on whether you're going to use just the plastic dishes as they kind of come from the company that are tissue culture treated, or whether you need to add something on top of that. Okay, so now that I've sprayed all of those in, I'm also going to open them before I get started, just so I don't have to open them kind of as I go. So when I open them, I'm going to unscrew them, and then I'm going to leave the lids loose, setting on top. Just so they're easy to open while I'm working. And so I've also set this up in a specific organization where I have my media on one side and the pipettes on the other, leaving me a space to work with my cell culture dishes that has a full view from front to back. It's important to keep in mind that things on the grate in the front, this is a non-sterile field. Here, where everything between that front grate and the back is going to be sterile. And I want a clean flow of air kind of going front to back for where I want my cells to be to maintain the most sterile environment. Before I take my cells out of the incubator, because I am working with primary cells today, I need to treat my future dishes with this gelatin mixture and let them sit or incubate for a certain period of time. Again, this may or may not be necessary on what you want to do, and that incubation time may vary, but for my protocol, I need to incubate the dishes with gelatin for 30 minutes. So I want to do that now before I get the cells out, because the rest of my protocol takes less than 30 minutes. So I'm going to take my cell culture dishes. You see they are taped. The inside is going to be sterile, and so I want to open them inside the incubator, inside the hood, sorry. And then I'm going to pull out however many dishes that I need. Today I'm going to be pulling out 3 different dishes. So then, to maintain the sterility of the pack of cell culture dishes, they need to be closed, also inside the hood here. So you can see I'm not setting them on any sort of surface here. I did not spray them down. I'm just holding them inside there and making sure they don't touch anything else. So now I have 3 dishes and I've moved them towards the back, because that's going to be where my cells end up in the future here. So when I open dishes in the hood, I always want to open them so that the open part is facing as far back as possible, because the back is the most sterile area. I've taken a 5 ml pipette here, and I'm going to add 5 ml of our 1% gelatin solution into each one of those dishes. So to do that, I'm going to push the end of the pipette through the paper back, not the plastic, the paper back of the pipette, and put it into the pipette men, so I can no longer see the little cotton swab here at the end. And then I'm going to pull off that, and that goes into the trash. And I'm going to be very careful of where this goes, because if this pipette tip touches anything like the walls or the floor of the hood or the glass towards my face, I'm going to need to get a new one. It's no longer considered sterile at that point. But because I've already opened the lids here, it's just easy for me to open them, and I pull up 5 ml of that gelatin solution. And because I've already opened my dishes towards the back, I can just squirt that into each dish. Now there are no cells involved here, so I'm going to use the same pipette for all 3 of them. 1 pipette per solution of liquid. So I'm not going to use the same pipette for any of the other medias or any other things. This one was just used for gelatin. And that's going to go into our biohazard waste. So these ones, because the dish of the cells is fairly hydrophobic, it's not going to immediately disperse over the surface. So I'm just going to rock them gently back and forth to make sure that gelatin liquid covers the entire bottom surface of the dishes. And then we're going to let that sit for 30 minutes while we do the rest of our cell culture passing. So now I'm going to pull my cells out of the incubator. Just like you checked your media for any sort of contaminations, you should do that with your dish of cells as well to make sure there's no opaqueness or floaters in them. You may want to check them underneath a microscope, which you can see one of the other videos for before you get started here. I'm not going to spray down the dish of cells because they should be going from the sterile environment of the incubator to the sterile environment of the hood. So we're going to do 3 kind of routine steps to pass our cells. This includes taking the media off of the cells that is currently in that dish. So these are adherent cells, meaning that they're stuck to the bottom of the dish. So we're going to take the media that's going to leave the cells stuck on the bottom. We're going to wash those cells with PBS once. We're going to take off that PBS. We're going to add trypsin to them. The trypsin is an enzyme that's going to cleave the connections between the cell and the dish. So it's going to put the cells into suspension that are going to be floating in that trypsin solution after a certain period of time. And then we're going to mix new media with that trypsin solution to move those cells from this one dish and then split them into these 3 dishes that are currently incubating with gelatin. So those are the steps that I'm going to go through. This is our aspirating system. It's just essentially a vacuum pump with a little gun attached to it. I'm going to put a pipette tip in there. Yours may look different, but it essentially is going to be some sort of vacuum system that allows you to pull the media off and then collects it for you. Like I said, ours uses the glass Pasture pipettes. Yours may use something different, but whatever you use for your vacuum system. So when I open my dishes, I'm not going to take the lid off and set them off to the side or anything like that. I'm just going to tilt them and open it towards the back. Again, the back of the hood is going to be the most sterile environment. And then I am just going to suck up that liquid and aspirate off that media. Because these are glass, they're going to go in a specific biohazard glass waste. Everything, all the pipette tips we use from now on, they have touched something with our cells in it that makes them biohazard waste that needs to be collected and disposed of properly. Now I'm going to wash that dish of cells with 5 mils of PBS. Again, I tore through the back to open that pipette, put it in the pipette bin. So that's going to be our PBS. What the PBS does is that it helps the trypsin to work faster. The trypsin will work without it, but doing a PBS wash just kind of speeds that process up. So it doesn't need to really incubate for any period of time. You can do a couple rocks back and forth, but you just put the 5 mils of PBS on, and then we're going to aspirate it off. Now we're going to add 2 mils of the trypsin solution. Again, that's going to be the enzyme that removes the cells from the adherent dish. And then we're going to incubate that trypsin solution back into our 37-degree incubator for 5 minutes to 15 minutes. It depends on what cell type you're looking at. And you'll want to check underneath the microscope to make sure you can see your cells all floating before you proceed after that point. So I'm using a 5 mil pipette. There are specific 2 mil pipettes, if you have them, that you may use. And then we're just going to make sure it's covered on the bottom, and we're going to put that back in the incubator. So now it's been 5 minutes that our cells have been trypsinizing, and I've checked them to make sure that they're all floating on the microscope. Again, you can see the other video of how to look at your cells underneath the microscope. So I'm going to take them out of the incubator and add the media and get them split into our 3 different dishes. So now the cells are floating in the 2 mils of trypsin liquid that are in this dish. So it's really important that you do not aspirate off this media, because then you're going to lose all of your cells. You need to add your growth media to this to deactivate the trypsin from continuing to chew on the cells and to create enough liquid for you to split into our different dishes. So earlier we set up those 3 gelatin-coated dishes. So they've been sitting now for 30 minutes. So as I pull out the cells with the trypsin, I also don't want to be too laggy on this because, again, the trypsin continues to activate and kind of work on those cells. I'm going to aspirate off the gelatin mixture from those plates so they're ready to go, and then I'm going to add 8 mils of growth media to those 2 mils of trypsin, creating 10 mils of a cell solution in total. And I'm going to spray my hands with ethanol before I get started. Okay, so I've added 8 mils of the growth media to the dish, so that brings our total up to 10 mils. 8 mils of growth media, 2 mils of trypsin makes 10. So the trypsin should be lifting the cells up off the surface, but you do want to do a couple washes of the dish bottom. So I'm just pulling up those 10 mils and then spraying it back out while the dish is tilted to really just kind of wash that surface here. So do that like 2 to 3 times, and then I'm going to pull up all 10 mils into my pipette. And so this is going to be a 1 to 3 passage or split of the cells, because I have these 3 dishes. So I'm just going to split these 10 mils evenly between those 3. So really that's 3 mils and a little bit to split out those 10 mils. And these again are open to the back, so I can just place those 3 mils into that open space, hopefully not spilling any on the surface. And now I can get rid of my old dish, but you might want to keep it if you don't remember the things that are written on it, such as your cell name, your passage number, your date. Those are things that are going to have to be written on your new dishes here in a second. But otherwise, it is biosafety waste at that point. So in our new dishes here, these all have about 3 mils in them that contain cells. Then I want to add 7 mils of new growth media to each one of those to bring the volume up to 10 mils total in these dishes. The amount of media total in your dish is going to depend on the size of your dish, and then it can also change whether you're using tissue culture flasks or not versus dishes. So I am going to grab 3 10 ml pipettes here because I'm going to use 1 pipette tip per dish for this process. The reason for that then is because when I pull up those 7 mils from the media bottle and then put those 7 mils in the dish, that dish has cells in it. So I don't want to use the same pipette tip to go back into my bottle, and that can possibly contaminate my bottle of media with cell solution. So 1 pipette tip per dish. And you never want to go from something that has cells back into something that does not have any cells in it. And then same kind of process as we did with the gelatin, you just want to gently rock them so that the liquid is covering the entire surface. The gelatin should make it so it's not nearly as hydrophobic as it once was. So then I want to label the outside edges of my dish here with my cell name, my initials, the date, and then the passage number. So these are going to be passage 3 for us. And then also the ratio that I use. So this is going to be a 1:3, 1 to 3 split. And you want to label all of them the same. Having them labeled on the outside is important for, again, looking at them underneath a microscope. So now that the cells are passed, we're going to put them back into the incubator. How often you need to do this passage really depends on the confluency of the cells in the dish. Again, something we talk about when we talk about looking at your cells underneath a microscope. You don't want them to get overgrown. So a lot of standard human or mouse cell cultures would be 3 to 4 days-ish, but it's really dependent on your cell type and monitoring them closely.